Hello everyone. Today we are going to discuss a very important topic, the bacterial cell wall. First of all, we should know about the structure of peptidoglycan. As we can see in the picture, it is formed by a carbohydrate backbone composed by alternating N-acetyl glucosamine and N-acetyl muramic acid. Both of them are linked through beta 1,4 linkage. This beta 1,4 linkage is the target of enzyme lysozyme, which is found in lots of secretions of the human body like tear, saliva, gastric secretion, etc. There is a set of tetrapeptide side chain which is unique to each bacterial species. Like the first one may be the L-alanine, second one may be D-glutamate or substituted D-glutamate. Third one is the most variable one. In case of gram-positive bacteria, it is L-lysine, and in case of gram-negative bacteria, it is mostly diaminopimelic acid. The fourth amino acid of this tetrapeptide side chain is the D-alanine, which is the most surest one among these four tetrapeptides. Then there is a cross link between these tetrapeptide side chains, and these cross links varies a lot between the species. For example, in Staphylococcal aureus, 5 glycines link the terminal D-alanine to the penultimate L-lysine. Based on Gram staining, all the bacteria are divided into two major groups. First one is the gram-positive bacteria and the second one is the gram-negative bacteria. There is another criteria which are the acid-fast bacilli. So first of all, let's have a look at the gram-positive cell wall. As we can see in the picture, in the gram-positive bacterial cell wall, the peptidoglycan layer is very thick, almost 40 to 80 sheets of peptidoglycan, where in case of gram-negative bacteria there is only 1 to 2 sheets. The gram-positive bacteria has teichoic acids, which is a very important characteristic of a gram-positive bacterial cell wall. Teichoic acid is actually a polymer of ribitol phosphate or the glycerol phosphate. As we can see from the picture, there are actually two types of teichoic acid. One is the wall teichoic acid, which is actually made of the ribitol phosphate and attached to the peptidoglycan layer. Another is the membrane teichoic acid or the lipoteichoic acid, which are actually composed of glycerol phosphate. These are the lipoteichoic acid. And as we see, there are no periplasmic space in case of a gram-positive bacteria, which is a characteristic of this type of cell wall. Now it's time for the gram-negative bacterial cell wall. In this picture, we can see there is a very thin peptidoglycan layer, which was in case of gram-positive is very thick. In this case, is only 1 to 2 sheets of peptidoglycan. Second one, there is an outer membrane which has an inner leaflet made up of phospholipid and an outer leaflet which contains lipopolysaccharide. This lipopolysaccharide is made of three parts. The first part is a lipid A, second part is a core polysaccharide, and the third part is the outer O antigen. But most of the bacteria have lipopolysaccharides, true, but some bacteria have lipo-oligosaccharide. For example, Neisseria meningitidis, Neisseria gonorrhoeae, Haemophilus ducreyi, they all have lipo-oligosaccharide instead of lipopolysaccharide. Now let's have a look why LPS is also known as endotoxin. First of all, there is a part of lipopolysaccharide is lipid A, which is actually responsible for its toxic effect in the living organisms. And there are two types of toxins from the bacteria: exotoxins, which are actually not part of bacteria and actively secreted and the endotoxin, which is lipopolysaccharide, which are not actively secreted and which are the part of the bacterial cell wall. So that's why lipopolysaccharide is an endotoxin, not an exotoxin. Now the core polysaccharide is made of 25 repeating units of sugar. 3 to 5 types of sugar are there present in these repeated units. And after the core polysaccharide, let's have a look at the O antigen. The O antigen means outer antigen, is also known as cell wall antigen or the somatic antigen. We can see there are 1500 serotypes of Salmonella, 150 serotypes of E. coli based on this O antigen, and against this antigen the antibody is made is always IgM type, as this O antigen is actually a polysaccharide, not a protein. So only and only IgM antibody will be made against this O antigen. Is a very important basic of immunology which is applied to this cell wall structure. There are porine proteins in this bacterial cell wall, because as it is a very complex structure in case of gram negative, so for the sake of the transport of very essential elements we need porine proteins here, but gram positive bacteria do not have any porine proteins. Now periplasm, which was not present in the gram positive bacteria. Let's have a look at the contents of periplasm. First of all, as we can see, there is this very tiny little sheet of peptidoglycan layer present in the periplasm. Then there are some proteins, and third there are the beta lactamase enzyme which breaks down the beta lactam antibiotics. Example of which is penicillin. And lastly there are some hydrolytic enzymes like alkaline phosphatase 5' nucleotide present in this periplasmic space. And as we see, there are no teichoic acids, Now we will have a look at the cell wall of acid first bacilli. They have a very unique structure which is mycolic acid composed of 70 to 90 carbons, which are linked to arabinogalactan, and as we can see there are very small amount of peptidoglycan in their cell wall. Mostly we think of Mycobacterium tuberculosis and Mycobacterium leprae are the acid first bacilli, as they have very high content of mycolic acid, but other bacteria like Nocardia and Corynebacterium diphtheria are also acid first bacilli. Now there are the last topic of today's is we should know what is protoplast, what is spheroplast, what is L-form, as it is related to the structure of a bacteria. First of all, what is a protoplast? In an isoosmotic environment, if we apply penicillin or lysozyme, most of the peptidoglycan layer of the gram-positive bacteria is washed out, which is then called the protoplast. And same treatment in an isoosmotic environment, if we apply lysozyme, EDTA, then huge portion of the gram-negative cell wall will be washed out, but they will retain their outer membrane, and in the outer membrane there will be entrapped peptidoglycan, and this form is called the spheroplast. And if protoplast and spheroplast are allowed to grow and divide, then it is called the L form of bacteria. So this was a protoplast, spheroplast and L form. So one trivia question: does every bacteria have cell wall in this world? No, the answer is no. Every bacteria do not have cell wall. The exception is Mycoplasma. They are actually the smallest kind of bacteria found in the world, and they do not have any cell wall. In fact, they have the very characteristic of cell membrane which contains high amount of sterol, very unique characteristic in this bacteria. So if you like the video, don't forget to share and subscribe our channel, and don't forget to hit the like button if you like this video. So thank you all, thank you very much.